This is the second podcast in our series on connective tissue. And here I'd like to look at some components of connective tissue, and we can talk about the matrix and the cells of connective tissue. The matrix, or ECM, extracellular matrix, consists of fibers, essentially two types of fibers: collagen fibers, which are the most abundant fibers in connective tissue, and elastic fibers. Collagen fibers are actually the most abundant structural component in connective tissue. Collagen is the most abundant protein in the body. Elastic fibers, as you might imagine, allow connective tissues to respond to stretch and to distension. And then the ground substance in the extracellular matrix consists of several types of molecules. We've got proteoglycans, so for example molecules like aggrecan or syndacans are proteoglycans. There are various adhesive proteins like laminins and fibronectins, and then there are various glycosaminoglycans like dermatan sulfate or keratin sulfate or hyaluronic acid. And then of course the cells of connective tissue, there are resident cells and transient cells. Resident cells are the so-called stationary cells in connective tissue. These would be cells like fibroblasts, maybe myoblasts, macrophages, mast cells, adipocytes, and even adult stem cells. And we'll consider some of the resident cells later. And then there are transient cells or wandering cells. These are essentially cells that come into connective tissue from the peripheral blood. So cells like lymphocytes, plasma cells, neutrophils, eosinophils, perhaps basophils, and monocytes. We're not going to consider the transient cells of connective tissue in this series of podcasts because we're going to talk about them when we talk about blood cells in a later portion of the course. Now, the fibrous proteins of connective tissue, the extracellular matrix, the collagens, these are non-distensible proteins. They give the connective tissue great tensile strength, and of course the elastins are very highly distensible, so they give the connective tissue some recoil capability. As you probably remember from biochemistry, collagen molecules are essentially composed of a triple helix. The triple helix makes up the basic structural unit of collagen. Collagen fibrils are essentially assemblies of tropocollagen molecules, and the fibrils make up collagen fibers. A collagen fiber is essentially a bundle of collagen fibrils. Now, as we said, the collagen is not distensible, so it gives the connective tissue great tensile strength. On the other hand, elastin is very highly distensible, and there are two components to elastin fibers. That is an amorphous component, which is elastin proper, and there's a fibrillar component, which is called fibrillin. Collagen fibers, as we said, are the most abundant structural component of connective tissue. Collagen fibrils have a subunit structure. The subunit structure of collagen fibers are what's observed with the transmission electron microscope. These little fine individual fibers are the individual collagen fibrils. Here they're shown at a little bit higher magnification. And here shown nicely in the scanning electron microscope. So this would be a collagen fiber. Here are the individual fibrils. And here you can see by transmission microscopy the little bit higher power of the collagen fibrils. And what's very apparent, there's a distinct banding pattern that you see in the collagen fibrils. This banding pattern repeats about every 68 nanometers. I don't care that you remember that. This reflects the fibrils' molecular structure and arrangement of the collagen molecules. And it turns out that each collagen molecule, you may have heard the term tropocollagen from biochemistry, each collagen molecule consists of 3 polypeptides that are known as alpha chains, and these alpha chains wrap around each other in a triple helix. Every third amino acid in an alpha chain is a glycine, and each glycine is usually preceded by a hydroxyproline, and it's usually followed by a proline. Now, I'm not going to go over this slide. You should review this from biochemistry studies, but you've talked about this in biochemistry. We've talked about it in our course earlier in relation to just some of the basic functions that occur with protein synthesis in eukaryotic cells. So you could look at the synthesis and assembly of collagen fibrils and relate that to the cell biology. And that's what this slide does. And I would like you to review this information from your biochemistry course. But, for example, you know about gene transcription, you know about splicing the original mRNAs, you know about translocation of the mature mRNA into the cytoplasm, where it will be translated on membrane-bound ribosomes. You know about proteins translocating across the ER membrane. They're processed in the ER membrane, packaged in the Golgi complex, inserted into secretory vesicles. The vesicles fuse with the plasma membrane, and pro-collagen molecules will be excreted from the cell. And of course, you should remember from your biochemistry, there's additional processing once the pro-collagen molecules are excreted from the cell, and the final collagens assemble actually in the extracellular matrix. There are at least 25 different collagens. I think your textbook now suggests there are 28 different collagens. There are at least 40 different types of alpha chains. These are encoded by genes on different chromosomes. For the point of our course, I want you to know about type 1, type 2, type 3, and type 4 collagen. So I've set up this little table to help you understand at least type 1, 2, 3, and 4 collagens. Now, I don't care so much that you know the composition of the different collagen molecules. I put this component of the table in to reiterate what you've learned in biochemistry. I do want you to know, for example, the type 1 collagen is found in bone and in tendon, and, for example, in ligaments, in the dermis of the skin, in the sclera. It turns out the type 1 collagen consists of about 90% of all the collagens in the body, and type 1 collagen gives the organs and tissues resistance to force with tension and stretch. Type 2 collagen, hyaline cartilage, elastic cartilage. You find it in the notochord and in intervertebral discs. Resistance to pressure. Type 3 collagen, loose connective tissue, and in a variety of organs like the uterus, liver, spleen, kidney, lung. You may find it in smooth muscle, in the endoneurium, in blood vessels, and even in the skin of fetus. Type 3 collagen forms these very loose reticular fibers. It forms a loose stromal scaffolding in which the parenchymal cells of many organs are interdigitated. So that's type 3 collagen. Type 4 collagen, basal lamina, primarily basal lamina of epithelium. It's also found in the lens capsule in the eye, and it's also found in the glomeruli in the kidney. Major functions of type 4 collagen for support, and when we talk about the kidney, we'll talk about it as a major component of the filtration barrier in the kidney. So as I said, I want you to know about these 4 types of collagens for our course. You will encounter the other collagens later in your clinical training. If you talk about certain disease conditions for collagens, you may talk about some of the other collagens. The image on the top left shows collagen 1 fibers in the dermis of the skin. Notice how thick and dense they are compared to the thin collagen type 3 fibers, the so-called reticular fibers that you find in a lymph node. So here you can see the very thin reticular fibers making up the stromal network in the lymph node, and contrast that to these very thick collagen fibers in the dermis. There are a variety of collagenopathies, basically mutations in collagen molecules, that you're going to talk about and you'll hear in your clinical training. Most collagenopathies are considered dominant mutations, and a general rule of thumb in genetics, if you have a structural mutation, like a mutation in a structural protein that leads to an obvious structural defect in an individual, those are usually dominant mutations, and most of the time they're autosomal dominant mutations. So there are 4 major collagenopathies that you might run into even earlier than your clinical training. For example, osteogenesis imperfecta defecta. It's an autosomal dominant mutation in collagen type 1. Individuals with this mutation have brittle bones, weak tendons, fairly thin skin. They have abnormalities in their dental structures. Their sclera are actually blue, and that's because the sclera are thinner because there's less functional collagen type 1 in their sclera. That allows the underlying tissues to show through. Kinase dysplasia is an autosomal dominant mutation in collagen type 2. Those individuals usually have short stature, restricted joint mobility, and they have vision problems, and oftentimes they're blind. There's an autosomal dominant mutation in collagen type 3 that causes Ehlers-Danlos syndrome. These individuals have hypermobility of the joints, and they have blood vessel problems. Oftentimes they'll have aortic dissections or other blood vessels that just rupture. And then collagen type 4 mutations, Alport syndrome. This one turns out to be an X-linked dominant mutation. These individuals have filtration problems. They have hematuria, that is blood in the urine. They don't have proper filtration at the glomerular basement membrane. They also have ocular issues and progressive loss of hearing. Let's talk briefly about elastic fibers. There are two components to elastic fibers. There's elastin itself, which is an amorphous protein. About a third of the amino acids are glycine, perhaps another 10 or 11% proline. Elastin has an abundant amount of hydrophobic amino acids. There are also some specialized amino acids, desmosine and isodesmosine. This helps to give the elastic fiber some of their unique stretching capability. And then tetramers of lysine help to cross-link adjacent elastic fibers. And then the fibrilla component of elastin, fibrillin, is actually a fibrilloglycoprotein. This protein stabilizes the amorphous elastin molecules, and you'll hear about Marfan syndrome later. That's an autosomal dominant mutation where there's mutations in the fibrillar gene. Now, elastin is synthesized primarily by fibroblasts and also by vascular smooth muscle cells. You're going to understand that some blood vessels have a lot of elastic fibers associated with them, and those fibers would be synthesized by the smooth muscle cells. Elastic fibers give tissues a distensibility with a recoil, and if you think of what would happen in an elastic artery like the aorta, of course there's a lot of distensibility and recoil with systole and diastole. Elastic fibers tend to be very, very stable. They have a very low turnover rate. Here's just a histological image with the thin squiggly structures, which are the elastic fibers. The thicker components here are fibers of the collagen fibers. Here's a diagrammatic view of the elastic fibers showing this desmosine, one of the specialized amino acids, and then lysine tetramers that help to hold the elastic molecules together. And then the diagrammatic view just shows the ability of the elastic molecules to really stretch out and then with a recoil to relax. And you can imagine this ability of stretching and recoiling, very important for the physiologic function of some of the major blood vessels. Ground substance is that portion of the extracellular matrix that occupies spaces between cells and fibers. So let me just try to put in perspective what this cartoon is showing. These large pink structures would be typical collagen 1 molecules. And then you can imagine, although they're not shown in the cartoon, but perhaps there are various cell types of the connective tissue that would be associated with these fibers. But then the ground substance would be these molecules that are taking up the space between the collagen fibers and the cells that are in connective tissue. The ground substance is usually a viscous substance. It's clear. It has a high water content. In routine histological sections, ground substance just appears as empty space. As you might imagine, the ground substance consists of large proteoglycan core molecules to which abound various glycosaminoglycans and adhesive proteins. So here, just showing diagrammatically, maybe a core protein with these glycosaminoglycans linked with linker proteins onto a hyaluronic acid. Notice it almost looks like there's a bottle brush appearance to this ground substance. And in fact, this ground substance does have this bottle brush type appearance at the molecular level. There's a lot of negative charges on this material in the ground substance. This ground substance can actually hold a lot of water. The major components of ground substance, as we said, glycosaminoglycans, proteoglycans, and various glycoproteins. The glycosaminoglycans are also called mucopolysaccharides. These are polysaccharides with repeating disaccharide units. The basic unit is a hexose or a hexuronic acid linked to a hexosamine. Common ones are chondroitin sulfate, keratin sulfate, dermatan sulfate, heparan sulfate. Hyaluronic acid is one of the very largest of the glycosaminoglycans. This one exists only as a free glycosaminoglycan. Then proteoglycans themselves consist of a core protein and at least one O-linked glycosaminoglycan. These can exist as monomers or as aggregates bound to hyaluronic acid. Then some of the glycoproteins, adhesive proteins, which can bind calcium. So you've heard of molecules like fibronectin or laminin, osteopontin, for example. Now it turns out, and this is an important point, connective tissue cells secrete different ratios of these molecules in the ground substance. So that's the extracellular matrix is going to possess specific mechanical and biochemical characteristics that are going to be unique for the tissue in which it is found or in which it is present. And these cartoons just show some diagrammatic views of proteoglycans on the left and adhesive glycoproteins on the right. So some of the proteoglycans, aggrecan is important for hydration in cartilage. Virsican is important in cell-to-cell binding and cell-to-extracellular matrix binding. I'm not going to ask you to worry that much over the molecular details of these structures. I just want you to get a sense of how they're put together. Decorin orients collagen fibers, and then syndactin binds the extracellular matrix components to cytoskeletal components within embryos. We've already talked about fibronectin in many tissues and laminin within epithelial cells, muscle cells, adipocytes, and Schwann cells. And you can remember from our discussions on epithelium, these fibronectins and laminins help to bind the epithelial cells to the underlying basement membrane, to the connective tissue itself. Tenacin is an adhesive glycoprotein found in embryos. Osteopontin, of course, is important in binding to osteoclasts. Now, some important glycosaminoglycans to know about. Chondroitin sulfate, found primarily in cartilage and bone. I'm not going to ask you to remember the molecular structures here, but this will help you recapitulate from biochemistry. Heparan sulfate, found in the basal lamina and in loose connective tissue. Keratin sulfate, found in the dermis and in bone. Dermatan sulfate, found in the dermis itself. Hyaluronic acid, a very large molecule found in many different types of connective tissue. This glycosaminoglycan, as we said, it's probably one of the very largest of the glycosaminoglycans, and unlike the other ones I've mentioned, hyaluronic acid is not sulfated. This cartoon just gives you an idea of how the ground substance is assembled. So here is one of these core proteins, various glycosaminoglycans. They come together to form a proteoglycan monomer. Notice this almost like a bottle brush type appearance of this monomer. Then along with link proteins that will bind these proteoglycan monomers to the hyaluronic acid, you get this giant proteoglycan polymer. Lots of negative charges around here. This gives the connective tissue some resistance to being compressed. It will help to hold a lot of water in the connective tissue. So functions of extracellular matrix, as you would imagine. The collagen fibers give the connective tissue great tensile strength. The elastic fibers give the connective tissue the ability to stretch and to recoil. And the ground substance in the extracellular matrix allows the tissue to resist compression. It can resist penetration, allows the tissue to retain water and salts, and it also allows transport by diffusion.